Okay, also I'd like to thank organizing committee. And today I'll talk about the development of genome-edited chickens for egg component regulation and the production of recombinant proteins. The avian genome editing system is quite different from the mammalian genome editing systems. So in the mammalian case, microinjection of guide RNA and Cas9 into embryo or stem cell mediated genome editing method widely used for the genome editing. Also, SCNT method also frequently used for the precise genome editing. However, the avian system is quite different, and the microinjection or the stem cell mediated method cannot be applied in the avian systems due to developmental physiology. Therefore, germ cell transplantation and germline chimeric production method are widely applied in the avian species for the genome editing. Especially these primordial germ cells have received the most attention for the precise genome editing in avian species. Firstly, we can culture these primordial germ cells for the genome editing, and then genome edited primordial germ cell can transplant it in the blood vessel of developing embryo to produce germline chimera, and then we can confirm the generation of transgenic or genome edited birds. Also, the other method is we can direct inject bloodstream cell by the retroviral systems or lentiviral systems to direct modify in the bloodstream cells to make generate the transgenic or genome edited birds. So germline transmission system by using primordial germ cells made it possible to do precise genome editing in avian species. And then recently the McGrew group actually developed the SIODEM surrogate systems, and then this system so much improved the germline transmission efficiency. And then CRISPR-Cas system as a genetic scissor is also the most efficient toolkit for the precise genome editing. Yesterday, Alison already discussed this part. So, and then we can actually modulate or remove egg components by the genome editing method, the ovalbumin, over transparent, or over mucoid, lysozyme. This kind of the protein is major protein for the egg white, and sometimes it can be allergen to some people. So ovomucoid is the most allergenic, and ovalbumin is most abundant proteins in the egg white. So about 54% of egg white proteins. So we can remove or reduce this kind of proteins by using genome editing technology. Also, we can produce the recombinant protein in egg white or egg yolk. Firstly, we produced the ovalbumin knockout chickens by using Talem system, but at the time the progeny was not survived during the growing process. So we established another line by using CRISPR systems. We produced the ovalbumin knockout heterozygous chicken, and the mating of the male and female, we produced the ovalbumin homozygous knockout chickens successfully. And then this ovalbumin homozygous knockout chicken did not produce any ovalbumin in the oviduct of magnum area. So we confirmed in the gel, this is a wild type. They produced so much ovalbumin and over-transparent, over-mucoid in here, lysozyme. However, ovalbumin homozygous knockout chicken did not produce any ovalbumin proteins. And then these chickens come from produced from the ovalbumin homozygous knockout chickens. These ovalbumin homozygous knockout chickens were well grown until the sexual maturity without any abnormality, and they normally produce the sperm and laid eggs. However, the fertility was so low compared to the heterozygous ovalbumin knockout chicken, and also hatchability was just 34%. So when we compared to this heterozygous knockout chicken, this homozygous knockout chicken, hatchability was so low. So I think that means ovalbumin deficient eggs cannot fully support embryonic development. And then we figure out the concentration of the major proteins in the ovalbumin knockout, homozygous ovalbumin knockout chicken. This black bar is the ovalbumin homozygous knockout chicken, and this middle is ovalbumin heterozygous knockout, and this wild type. So over-transparent and the lysozyme and over-mucoid concentration tend to increase in the ovalbumin homozygous knockout chicken. But the egg weight and egg white volume decreased in the ovalbumin homozygous knockout chicken, but the egg yolk size was almost the same in the ovalbumin knockout chicken or heterozygous, homozygous, or wild type. And then for the precise genome editing, we tried to target nucleotide substitution by base editing technology. David Liu group firstly reported the base editing technology. They used the TED-Cas9. It's a kind of a mutated Cas9 that does not have a nuclease activity. So they connected the TED-Cas9 with some the cysteine deaminase or adenosine deaminase to convert the base pair. Dr. Kyung-Hyun worked for this project, and then we tried to induce premature stop codon in exon 2 of over-transparent genes. So we designed this guide RNA, and we targeted exon 2 this area because they include the TGG site. So we can convert the G to A to induce the stop codon TAA, TAG. So this G is 19th position of G in the middle, and the 20th G. So the efficiency of desired nucleotide substitution was about 80% in TF1 cell, but the pre-modial germ cells, actually base editing efficiency was not high. And then we produced about 80 proteins, and then we figured out the efficiency of desired substitution. 44 chicks out of 80 had the desired nucleotide substitution, but 22 chicks showed unwanted substitution. The nucleotide substituted in the different site, and then the 14 chick had a deletion mutation. So, and then the efficiency of base editing efficiency was highest at the G in the position of 19th. We tried to produce the homozygous over transparent knockout chicken, but this over transparent knockout homozygous knockout chicken showed embryonic lethality. The concentration of overtransferrin was about 60% of wild type. This is a wild type, and then this is a heterozygous overtransferrin and knockout chicken eggs. So we measured about 6 weeks, but the concentration was about 60% of wild type. Also, we can produce recombinant protein in the egg white and egg yolk. The egg yolk protein synthesized from the liver, and then the egg white protein synthesized in the magnum area of oviduct, and then deposited this area. After ovulation of this egg yolk, this oocyte, the added egg white in this magnum area. So Dr. Kim Yong-min and Ms. Shim Ji-yeon actually work for this Nagin project. We produced the recombinant protein in egg white. We inserted the EGFP gene into the ovalbumin locus, and then we want to disrupt ovalbumin genes, and then this EGFP gene, EGFP expression is controlled by endogenous ovalbumin promoter. So that's why this EGFP expressed in the magnum area of oviduct. And then this young chicken, they 17-old chickens, and they induced by the TS treatment as the estrogen analog. And then we GFP expressed in the magnum area. Also, this one is adult oviduct, and then also GFP expressed in the oviduct. And then GFP protein strongly expressed in the egg white. This is a wild type. This is a GFP Nagin chicken eggs. And then we confirm the GFP expression in the tweezers. So also strongly expressed in the oviduct. And the concentration of GFP was about 160 microgram per ml. And then also we tried to log in adiponectin gene into the ovalbumin locus, and then we also produced the adiponectin in the egg white. This is a control. This is egg white with adiponectin, and then the concentration of adiponectin was about 1.5 to 4.5 milligram per ml for the total adiponectin. So actually, so much adiponectin expressed in the egg white. That means we can produce about 30 to 9 milligram of adiponectin in one egg. So Dr. Kim Yong-min and Dr. Park Jin-se, they work for this project. And then ovalbumin, like egg white protein, also the recombinant protein from produced in the egg white usually showed the glycan pattern with high mannose and afucosylation and terminal galactosylation. So we produced the monoclonal antibody in the egg white, and then they showed so high the ADCC effect compared to the control, because this monoclonal antibody does not have the fucosylation. And then we also produced the recombinant protein in egg yolk. In this case, we tagged human immunoglobulin and FC genes right after albumin genes. So we connected with 2TA, and then we successfully produced the homozygous Nagin chicken. This one's a donor-derived progenies. And then this FC protein expressed in the liver specifically, because we use the albumin genes. And then this FC protein secreted in the serum and egg yolk. Also, we found the albumin and FC fusion protein in the serum. This is a non-reducing condition, as a reducing condition. And this one is homozygous FC Nagin chicken, male and female and male. So they were healthy, and then the concentration of FC protein was about 350 microgram per mil in the serum of homozygous Nagin chicken, and then in the egg yolk, the concentration was about 170 microgram per mil in the homozygous. So we can obtain about 3 to 5 milligram of FC protein in the one egg yolk. So as a conclusion, we can apply genome-edited chicken for the egg component regulation and the production of recombinant proteins in egg. So you can change egg composition, or you can reduce egg allergen. So we can produce the recombinant protein, egg white and egg yolk. That can be possible strong bioreactor systems. Also benefit for the glycosylation pattern. So mass production is possible. So, and I would like to thank my lab member here, Park Jin-se. He worked for the egg yolk bioreactor systems, and Dr. Choi and Dr. Lee Kyung-yeon, she worked for the base editing systems. And I would like to thank also my collaborator, Professor Hong Lee from Missouri, University of Missouri, and the Fulbright group, Dr. Daniel Nair and Professor Yong Yao and Professor Kim Tae-hyun. So funding from the National Research Foundation of Korea, and I'm running the Center for the Avian Germ Cell Modulation and Cloning, so funding from this institute. Thank you so much.